Hello, my name is Marom Bikson, and I'm going to be talking about why all forms of neuromodulation, all forms of brain stimulation can be thought of as subthreshold. So, the general notion behind all forms of neuromodulation, especially the kind that works by using electrical pulses, is that it activates the brain by generating action potential. So what that means, for example, is if we're applying 100 hertz stimulation to a particular part of the brain, we will then drive neurons in that particular part of the brain to fire at 100 hertz. We are generating action potentials, we are driving activity. So neuromodulation is a consequence of driving action potentials. However, here are a couple questions. When we're using neuromodulation, for example, to treat disease, how many patients present with not enough action potentials, right? How often is that the diagnosis when someone's suffering from a movement disorder or from pain, to say, oh, you're missing some action potentials, I'm going to give you some more action potentials, right? So that obviously doesn't make sense. And also, as we know from a neuroscience perspective, if everything is action potentials, if all brain processes are action potentials, and we're just dumping more action potentials into the system, why would that necessarily just treat the disease, you know, and not do something else, okay? So I think an answer to these questions comes in part from thinking about subthreshold neuromodulation. And to introduce what subthreshold neuromodulation is, I first have to start by defining what is supra-threshold neuromodulation. So most neuromodulation technology is conceived of as supra-threshold. It starts with a stimulator, it could be an IPG, it could be external, that is delivering pulses, right? So the recipe is pulses, and those pulses are driving action potentials, right? In the most simple way, in a one-to-one fashion. So click, action potential, click, action potential in a particular population of neurons. And whatever benefit we get from the neuromodulation derives from this pacing, right? From this driving. Okay. In the case of subthreshold neuromodulation, we still start with a stimulator, but in this case, it might be delivering pulses, it's not necessarily delivering pulses, but they're not at a sufficient intensity to produce action potentials. But they still affect the cells by moderately changing their membrane potential. And the way that membrane potential changes will track the waveform we apply. And it is this change in membrane potential and tone which then leads to the benefits. All right, so having laid that out, to understand how subthreshold neuromodulation works, to understand the mechanisms of subthreshold neuromodulation, we need to focus on what does it mean to polarize cells a little bit, but not enough to make them action potentials in a way that is meaningful for clinical benefits or behavioral changes. So I'm going to describe three general ideas for subthreshold neuromodulation, three neuromodulation mechanisms, right? They are stimulation of cells that don't generate action potentials, oscillations, and changing synaptic processing and plasticity. And I'll go through each one of those, starting with stimulation of cells that don't generate action potentials. Now, if you think about it, this is inherently subthreshold. If suprathreshold stimulation is stimulating to get action potentials, if you have a cell that can't generate an action potential, you're not going to do that. So when we're talking about stimulating glial cells, that is subthreshold. And also when we talk about endothelial cells, that's subthreshold. And I'm especially interested in endothelial cells. When we think about endothelial cells, we also think about the blood-brain barrier, because these are the cells that form a core part of the blood-brain barrier. And we've been looking at for a long time now at how neuromodulation or electrical stimulation can change the endothelial cells and therefore change the blood-brain barrier. Okay, and we've learned quite a few lessons from this. One of them is that when you apply energy, electricity into the body, that electricity is actually collected through the blood-brain barrier into the blood. The consequence of that is that the electric fields that are present across the blood-brain barrier can be hundreds of times, 400 times higher than the electric fields around the neuron. So there's a concentration effect. And this is something we've really considered with computational models. And as this current crosses from the brain into the blood, right, it drags with it water as a result of electroosmosis, and other solutes. So what's happening is not only are we pushing current across the blood-brain barrier, we are dragging things with it. This is a brain clearance mechanism, as well as a brain metabolite delivery mechanism. And we found out that when you do all these things, stimulate the blood-brain barrier, drag things across it, it triggers molecular, genetic expression changes in the blood-brain barrier, meaning that the blood-brain barrier itself can be a basis for plasticity, as well as these other functional changes. So right away we have one subthreshold mechanism. All right, another subthreshold mechanism has to do with modulation of ongoing oscillations. Okay, and this is again something I'll be presenting at a very high level. We and others have been studying this for decades. The cells in your brain, excitatory, inhibitory, they form a network, and they're never inert. Right? So when we think about stimulation of the nervous system, we have to always think about the fact that we are stimulating an active system. We're applying stimulation on top of this ongoing activity. And when we specifically think about oscillations or networks, what's happening is that these cells have organized themselves, right, into collections of neurons that are generating this rhythmic oscillatory activity. And we and others have shown that when you have a system like this, it becomes extremely sensitive to even very, very weak stimulation. This is not stimulation that is intense enough on its own to sort of pace action potentials, but it is quite intense enough in order to modulate these ongoing oscillations. And so this is one experimental result that I want to show you right here. So what you're looking at is data from a rat, a brain slice. These are its baseline oscillations. This is gamma in a rat. It's around 35 hertz. And at this time point here, we are applying stimulation at a low frequency, 2 hertz. And we're applying sinusoidal stimulation and then we turn it off. Now what you notice is that during the stimulation, the oscillations come and go, right? They're strengthened, they're weakened, they're strengthened, they're weakened. But what is the frequency of the oscillations? It's always at 35 hertz. So the point is the brain is generating the 35 hertz oscillations, right? That's an endogenous phenomenon. We are modulating this. We're not generating 2 hertz activity. We're modulating ongoing activity, right? If you were just pacing, then you would just produce 2 hertz, right? But we are modulating. So that's the essential idea with network effects. Finally, the third mechanism, as far as subthreshold mechanisms, is synaptic processing, okay? So as everyone is very familiar, neurons are basically information processors. Everything is a result of this processing: cognition, behavior, disease. You have individual neurons, they receive synapses from other neurons, and they process these neurons. And as I mentioned with the oscillations, we need to understand that these synapses are always active, right? They're not in a state where we start from 0 and now we stimulate on top of that. Okay. Now, in the case of supra-threshold stimulation, for example, when you stimulate these axons, you will also activate these synapses. So it's correct to say that supra-threshold stimulation must be stimulation of synapses as a result of orthodromic conduction, but it's like a pacing, right? You're stimulating the axons, you're sort of overriding the ongoing activity. Subthreshold stimulation works differently. Subthreshold stimulation is not sufficient to drive these action potentials, but it is sufficient to polarize the terminals of the synapses as well as to polarize the dendrites. And what that has the effect of is of modulating the gain of the synapse. We can make synapses stronger or weaker so that whatever activity is ongoing will affect the cell in a different way. All right, these are the 3 high-level subthreshold mechanisms. Okay, let's take a step back. How does neuromodulation work in general, right? Can we put that in a single cartoon? I think so. All neuromodulation works. You have a neuromodulation device that provides a certain waveform to certain electrodes. Now you pick a particular waveform and that is your dose. So the dose is your electric position, your peak amplitude, your waveform, and that dose is the only thing that arrives at the patient, right? That's what's being delivered to the patient. It's why we define it as the dose. and that determines the outcomes, all right? A very critical idea, however, is that each neuromodulation device, it's not shipped with one dose, right? You don't get a neuromodulation device that only provides one intensity, or only works with one electrode position, or only works with one frequency, right? There's many possible doses in the neuromodulation device, and we're picking one dose to apply for it. Who picks it? The caregiver, right? I would say that's the job of caregivers and doctors, right, in neuromodulation. You're picking the device to begin with, which is a set of doses, and then at any given time, you're picking one dose, right? You can only pick one at a time that will be applied to the patient. Now, there's another important piece of this that we have to add on to it, which is that obviously the first dose we try may not give us the outcomes that we want, right? You're trying to treat pain, it didn't work. You're trying to treat pain, it did work, but there were unacceptable side effects. So we're looking at clinical responses. We're also potentially looking at biomarkers, right? And we're using that information to update the dose. What does it mean to update the dose? Well, there's many potential doses built into the neuromodulation device. We're going to be picking a different one. And of course, you can repeat this loop and loop again. And I really want to focus on this dose, right? Which is the only thing that the patient is getting. There's a couple really important corollaries to this idea. One of those ideas is this: if you have two different devices, right? And one is made by company A and one is made by company B, but they both provide the exact same dose to the patient. Same electrode position and waveform and intensity. As far as the patient is concerned, they are identical devices, right? They may have other differences, but as far as the immediate effects on the person's neurophysiology, they're identical, right? The two devices, the same dose, same outcome. Now, the flip side of that is, right, that if you want to innovate and make better therapies, well, the dial that you need to turn, literally sometimes, is to come up with a better dose. But it's not just coming up with a better dose, because we know that these devices have many possible doses built into them already. It's coming up with better ways to pick the right dose, right? The rules to pick the dose. So neuromodulation devices never just ship as is. They always ship with dose instructions, and those must be considered collectively when we're thinking about these devices. All right, taking another step back, I want to talk about the gate control theory of pain, which many people are already familiar with. So I'll go through it briefly, as developed by Melzack and Wall in the 1960s. So Melzack and Wall conjectured that non-painful input, right, goes from the periphery to the brain along what they call sort of large diameter axons. And these could travel in a straight line from the periphery straight to the brain. Now, they also conjecture that there were separate nerves, and they call these smaller diameter nerves, and these were carrying painful information, right? Noxious information. However, they don't have a straight line to the brain. They conjecture that they need to go into the spinal cord, make a synapse onto cells that they called T cells, that then project onto the brain. Now, they added, so this is, and this by the way was an excitatory synapse. Now they added to this a very important additional element, that the large diameter axons, in addition to traveling straight to the brain, spit off a collateral, and that collateral, through a series of synapses, inhibited this pathway. Right? So this was the basic circuit of it. So what's happening here is that the activation of these large diameter axons is, through this green pathway, inhibiting the transmission of painful stimuli, right? So this was the notion of the pain. The pain gate is here, and I want you to keep in mind that the pain gate is a synaptic signal, all right? The pain gate is synapses. And this powerful idea also led to very specific translational conjecture, which is that if you apply electrical stimulation in such a way that activates specifically these large diameter axons, right? Well, one thing you might do is you might produce some tingling, but you would also suppress pain, right? This was the conjecture of Melzack and Wall. So you stimulate the large diameter axons, you produce tingling, paresthesia, but you also close the gate, all right? And again, the gate that you're closing is a synaptic gate. And here's what that gate looked like. So Wall was a neurophysiologist, right? He was recording from, in animal models, what he thought was the synaptic gate, a slow synaptic signal. Now, what was extremely powerful about this idea is that you could actually stimulate in different locations, right? You could stimulate peripherally. You could stimulate sort of non-invasively, like with TENS. You could stimulate percutaneously. You could stimulate over the spinal cord. You could even stimulate sort of upstream from the site because of the sort of antidromic conduction that you would see here. So there's a lot of potential locations to stimulate that led to the creation of all these different technologies that we're now familiar with. So if we now think about how neuromodulation works for pain, we have the same picture, but now we're going to say, well, the thing we're going to be tuning to is paresthesia, right? Paresthesia-based neuromodulation, because that's the thing that will get us to the right dose in order to produce the treatment. And it was also recognized that in addition to paresthesia, you could also record a neurophysiological signal called the eCAPS. Now, very briefly, right, when we apply stimulation to the spinal cord, we activate these superficial white matter tracts that then conduct the information up and down the spinal cord. And it's important to recognize, right, that the gate is here. So it's only after this activity comes into the spinal cord and activates a synaptic process do we actually produce the closing of the gate. Now, if you take another electrode and you use that to record the activity, you can record what's called an eCAP as this travels down, heading towards the gate, right? So this is the signal of eCAP that has now been translated into closed-looped systems. But I want to emphasize the eCAP is recording the superficial conduction. It's not really recording, it's not recording the gate itself, but the signal on the way to the gate. And we were wondering whether it would be possible to record a signal from inside the gray matter itself, right? A signal that may reflect activity within the dorsal horn, and therefore perhaps the signals that are closing the gate or the gate itself. And at least in a rodent model, we were able to do this. So we identified something that we now call eSAPS. These are much slower signals, and we were able to show in a rodent model that these certainly derive from inside the dorsal horn, and they reflect synaptic signals in the dorsal horn. And this was very compelling for us because, again, the original discovery of gate control theory was itself derived based on a synaptic slow signal. Okay, so this is where we started with, where we have this loop. We're using paresthesia or even perhaps eCAP-based, paresthesia-derived programming, right? But now I think we've opened up the possibility that perhaps, because the real goal was to close the gate, the synaptic gate, not necessarily produce paresthesia, right? It would be possible to close the gate without paresthesia, right? So paresthesia free. And so if we go back to our cartoon of the gate control theory of pain, we can ask the question here, okay, how would we do this without paresthesia? And I really see that there's sort of two ways. One is you stimulate what they call these large diameter axons, but you do it in such a way that doesn't produce paresthesia. Maybe the pattern of it activates these axons, closing the gate, but when that pattern is transmitted centrally, the brain doesn't recognize that as tingling. That's one possibility. Another one would be, well, why don't we just stimulate the gate directly, right? Why don't we apply electricity straight into the gray matter, the dorsal horn, stimulate right here, and somehow close the gate directly that way, right? Now, in order to do that, you sort of have to overcome a bit of a dogma in spinal cord stimulation, which was that for many people and for a long period of time, spinal cord stimulation was considered white matter stimulation, right? Why was that? Well, for one thing, if you look at the anatomy, the white matter is much closer to the electrode than the gray matter, and so anything that's closer to the electrode is going to be more likely to be stimulated. And in fact, people like Holzheimer and others developed computational models that confirm this idea. The current that was generated superficially in the white matter was much higher than the current that was then generated in the dorsal horn. And in fact, only the currents that were generated in the white matter were high enough to drive action potentials. So supra-threshold stimulation would be predicted to be happening here, right? So that led to this conjecture that spinal cord stimulation works by dorsal column, white matter stimulation. And this was supported through different things. The original work on spinal cord stimulation, the sort of the pivotal case study, actually called it dorsal column stimulation, though the field moved on with that, but because it was assumed to work that way based on gate control theory. It's also the case that there is situations where you could stimulate in the periphery and produce pain control, and there it's clearly white matter stimulation, we're near the gray matter, so that was consistent with dorsal column stimulation. And there's also this notion that effective therapy required paresthesia. But why was that? Well, you programmed based on paresthesia to produce effective therapy, and therefore effective therapy was associated with paresthesia. We know now that that was a bit of a circular argument. Okay, it literally is, right, for the reasons that I think is obvious. We now know that there is many classes of sub-paresthesia therapy. In fact, all of these were a bit circular in some ways as well, based on this notion of combining gate control theory with supra-threshold stimulation. So again, this is not new. This is no longer dogma in the field. And now I think we're much more open to ask the question, okay, well, could the currents that reach the dorsal horn that are well below or somewhat below the intensity needed to pacing, too low to produce action potentials, still modulate the dorsal horn? And of course, now we're going all the way back to subthreshold mechanisms, where we have decades of work showing that absolutely fields that are not sufficient in order to produce pacing can modulate it, right? So coming sort of full circle here, that's why I really wanted to talk about it in this way, okay? And what are those subthreshold mechanisms? We already described 3 of them to you, right? So we're not starting from scratch, right? There's this massive basis that we can build on. Okay, so we and others have been working on this notion that spinal cord stimulation may work, in fact, by influencing interneurons in the dorsal horn, interneurons that are specifically involved in gate control. It would influence them by polarizing them, by changing how they process synaptic activity. And again, I would refer you to this preprint that's in work and also work by others showing that clearly the fields that are produced, the currents that are produced in the gray matter produce levels of polarization in the millivolt range, which may seem small for supra-threshold, but they're really big for subthreshold. These are fields that are potent, all right, to modulate synaptic processing, to modulate synaptic efficacy, to modulate network processing. Okay, so what do I want you to take away with here? Is the first of all is that we have well-established mechanisms of subthreshold neuromodulation. These have been characterized for decades, and you can, as a first pass, classify them into these 3 categories: cells that don't generate action potentials, modulating ongoing oscillations, not generating de novo, and modulating ongoing synaptic activity. However, for a long time, subthreshold stimulation was neglected in technologies like spinal cord stimulation, deep brain stimulation, transcranial magnetic stimulation. In fact, most neuromodulation technologies, because these technologies were intense enough in order to produce pacing. And because they were, all the attention went there. It was assumed that that must be what they're doing, because that is what, that is one thing that they're doing. And I'm trying to show you that's not the case. In fact, what I want you to realize is that if you have a particular region where the currents are high enough to produce supra-threshold pacing, you have a much, much larger region where you have subthreshold mechanisms at play. And moreover, inside the region, even inside the region where you have supra-threshold mechanisms, you're also activating subthreshold mechanisms. So if you're pacing neurons, you're also affecting endothelial cells, right? If you're pacing neurons, you're also modulating oscillations. And so that's why sort of my thesis here, right, is that all neuromodulation, right, is subthreshold. Okay, thank you for your attention.